So we got our light boxes on. We've looked at our cultures from last week, right? Do you have isolated colonies? Most of you guys did. After about 24 hours of growth at 37 degrees, which is typically what we would do for E. coli, but I changed it up and we did room temperature because I wanted that other organism to turn pink. It actually produces a red pigment, but if there isn't a lot of the red pigment, it's a lighter shade of red, which we know as pink. So, how many cells do you think are in a colony if it started from a single cell? Like that little circle of growth that you see on that plate. About how many bacteria do you think is in that circle? 1,000, 1,000,000, billions. Potentially billions. In a liquid culture with aeration, E. coli can double itself every 20 minutes under those conditions. Yeah. Every 20 minutes, you go from 1 E. coli to 2. On a plate, it's a little bit slower, right? Access to nutrients isn't as good. So say it's 30 minutes on a nutrient agar plate. So after 24 hours, right, if every half an hour it doubles, that would give you 48 doublings. So that would be 2 to the 48th power. So that's 2 times 2, 48 times. So we could kind of shorthand it and say it's, 2 to the 50, right, to make it easy math. Do a guesstimation, right? 2 to the 10th power is, again, 1,024. We can round that to 1,000. Times that by 5, right? Would give you 1,000,000 times 1,000,000,000. That's a lot. Actual number, right, instead of 1 to the 15th, it'd be 2.8 times 10 to the 14th. Still a lot of bacteria. So we were off by, a factor of 3.5. Yeah. You don't have to know the math, right? But just to give you some idea, later on we're going to do this math. Not right now. Alright, so it really depends on the species. I like E. coli because, I could start a culture in the afternoon and have something the next morning, right? Because he grows so fast. He's awesome like that. Micrococcus luteus, on the other hand, is slow. It's very slow. I had to start that culture for you guys like a week in advance to have enough growth. And again, these cultures have been going for how long? A week. You did them last week, right? So, temperature also affects. We put E. coli at 25. It prefers 37, but it'll still grow at 25, just slower, right? So you got to know the conditions, temperature, things like oxygen availability. Can't do that. Okay. Sorry. So they kind of pigpile on top of each other, on top of that agar plate. So oxygen availability is different within the architecture of that colony. Whether they can grow aerobically or anaerobically with or without oxygen will really determine how they grow. Access to nutrients. So that shaking incubator where they're constantly mixed up and they're exposed to new nutrients and getting away from their waste in a broth culture is ideal when you want to grow large numbers of microorganisms and keep them happy, which we don't actually do in this lab. But so some scientists did an experiment to actually see, E. coli, and this is a link to that paper, about how many cells are in a colony, and on an agar plate there really is more like 3 billion instead of 100 billion. So probably doubling about every 45 minutes. That's still pretty fast. We can experimentally determine the number of, right? So we could do an experiment as well. In the past, we have done that. We are not going to do that this semester, though. So for Lab 4, we're going to use the cultures from last week, and we're going to make new cultures for next week. So the first thing that we're going to do is we're going to do what's referred to as subculturing. We're going to basically move them from their current vessels into a new fresh culture, right, that's sterile. New fallout shelter, right, for another week. So you'll have fresh cultures, so they'll stay nice and pretty for next week, and so that I don't have to do it. And it gives you guys practice. So we're going to start by doing some simple and negative staining. So we're just going to be using a single dye at a time and using 2 different techniques. So again, as promised, some terminology, but I'm not going to read through it. It'll be there for you guys. This terminology is some kind of really specific stuff that, yeah, we have you guys write out, but I'm not going to hold you to for testing or quizzing. Peter, one of the other instructors that teaches this course, and I are kind of in agreement that they get a little too insane, which is strange coming from him because he's all about molecules and how they interact. But what we really want to focus in on is that some of these dyes, actually all the dyes we use, have a charge to them. And that's how they're able to work, right? Because they have a charge component. And then also they have a color component, right? So for microscopy, important things and why we use microscopes is to increase resolution, right? How clearly we can see something. And we can calculate that for our microscopes, right? And this is a formula you do need to know, right? That we did do on the first quiz, and someone can close that door. What is the formula for resolution? What goes on the top? Wavelength. What do we measure wavelength in? Nanometers. What goes on the bottom? NA. What does NA stand for? It's one of your terms. You do need to be able to spell it out. Numerical aperture, right? You need the numerical apertures for what 2 lenses? The condenser and whatever objective lens you're using. Right? So we can calculate out the resolution. The next is magnification, which we're using a compound microscope, so total magnification is what times what? Oculars, which are set, times whatever objective you're using, right? That's pretty simple. So we want to be able to resolve them as separate entities, right? See them clearly. And we obviously need them magnified, otherwise we wouldn't see them with the naked eye. The third thing is contrast. And the type of microscopes that we're using don't create their own contrast. So we have to help them out. So what are we going to do today to help create contrast so that the bacteria don't blend in with their environment? We're going to stain them. So there are different types of light microscopes. Which type of microscopy do we use in our lab? Bright field, dark field, phase contrast, or fluorescence? Bright field. Where the background is bright, right? And the organisms are dark, mostly because we stain them. Dark field microscopy, the background is dark, and the organisms are illuminated. Phase contrast uses special filters that create contrast with adjusting the light. We don't have that either. And of course we don't have fluorescence microscopy, which utilizes fluorescent dyes in UV light microscope. So we use stains to create contrast so that we can see the organisms. The problem with staining, though, is it kills them, right? So if we need to see things like motility, we need to achieve that a little bit differently. And so after midterm, we will decrease the amount of light that we use in order to help us visualize them without staining them. And then, of course, we could use Serratia grown at 25, and it's actually pigmented, so it makes it a little bit easier to see it. So we can use them, and today we're going to use them basically just to see morphology, the individual shape and the arrangement of the cells. Other stains that we're going to use next week and the following week tell us a little bit more. So gram stain and acid-fast staining can tell us about the cell wall composition of the cells that we're looking at, in addition to being able to see the individual shape and arrangement because they've been stained. Endospore and capsule staining are going to help us see if they have an endospore or a capsule, right? Help us see those specialized structures. So these are known commonly as differential stains, right? They can differentiate one organism from another. So you're going to have one of two results. You're going to be gram-positive or gram-negative. You're going to be acid-fast or non-acid-fast. You're going to have a spore or not have a spore. You're going to have a capsule or not have a capsule. So I didn't see too many drawings, though, when I went around, but if you read on your homework, you were supposed to draw these. So I suggest going back and doing so, right, because you need to be able to identify and draw shapes of organisms. Remember, I don't collect your homework. This is for you, right? I don't analyze the quality of your drawing, right? It doesn't have to be beautiful. So some terms as it relates to the shapes of the organisms that we will see. And unfortunately for us in this lab, we only have two shapes. The first one, which is known as what? They're spherical, but what's the term we use for the spherical cells? Coccus, if you're talking about a single one. Cocci, if you're talking about all five of them in this picture. This is Latin. I is plural. The ending us is singular. It's kind of the opposite of what we're used to, right? When I say I, I'm talking about just me. When I say us, I'm talking about the group of us. This next one has them in pairs. What's the term for that? Those of you guys in lecture, this is a good review for the exam coming up next week. Starts with a D. This is Diplococcus. And again, that's if you're just talking about one pair. What would we say if we're talking about all of these pairs? Diplococci, right? Instead of the US, we put an I at the end. This is one of the arrangements we'll see today. It's groups of 4 known as Tetrads. This one is cocci in a chain, so this is known as Streptococcus, right? And this is the one that causes strep throat, right? Streptococcus pyogens. So when they're saying strep, they're just shortening the name of the organism that causes that infection. So I use that to my benefit. I think about strep throat. I think about how you don't want to swallow anything. You certainly wouldn't want to swallow a chain when you have strep throat, right? So I always think strep, I think a chain, I think about how I wouldn't want to swallow that. That's how I keep that separate from one of the other ones that students tend to confuse, which is coming up for F. E is a packet of 8 cells that forms a cube. This is known as Sarcinia. Yeah. The C sounds like an S because of the I, right? Sarcinia. The next one is Staphylococcus. And this is where you have the clustering. And we're going to see this one again today, too, because one of the organisms we're working with is called Staphylococcus Epidermis. So as you can tell by its name, it has this shape. And again, that's partly why it has that particular name. This gets confusing for students because some of these are not only arrangement names, right, this arrangement of clustering, but is also a genus name. And because of that, this next one, these are rod-shaped. right? The term is bacillus or bacilli, but a lot of scientists like myself, a lot of times just stick with saying rod-shaped because bacillus is a genus, a group of bacteria, like Bacillus anthracis. And so it gets confusing sometimes. Are you talking about shape or are you talking about that genus of organisms? Make sense? But you do need to know the proper terminology. So this next one are bacilli, right? But they're forming pairs. So pairs. Diplobacillus. We will see bacilli today. Two of the organisms we're working with are bacilli, are rod-shaped, E. coli, and I'm going to give you guys a new culture, Bacillus cereus. Well, that tells you right there. It's definitely rod-shaped, right? But that one actually will form chains. So you'll see them lining up in a chain. And so what's the term for chain? Strepto. So this would be Streptobacillus. And then, so we have cocci and rods in this lab, or bacillus. We don't have any vibrio. This is where it's a rod with a little bit of a curve to it. This particular picture, it actually shows flagella. And it's very common for this particular shape of organism to have flagella. And this is also a genus name. You may have heard of Vibrio cholera, right, which can cause a really bad infection of the intestines. This next one is a rod with multiple curves. This is known as spirillum or spirilia if you had more than one. And then also in this picture you see these lines at each end. That's denoting flagella. And again, a lot of the ones that are spiral shaped, spirillum, have flagella. This is actually all one organism. It doesn't hold a characteristic shape. This form is referred to as, anyone know? Pleomorphism. Pleo meaning many, morph referring to shape. Right? They're pleomorphic. The last one are the spirochetes. And so under the light microscope, they look like ziggy lines. These are different from the spirillum, which tend to have big curves to them. These guys, if you were to look at them, are actually helical, like a spiral staircase. And they have this characteristic shape because in their outer sheath, they have modified flagella called axil filaments that run along it, and it actually causes it to form this helical shape, and they can corkscrew through their environment. So they kind of spin through. So things like the causative agent of Lyme disease and syphilis are spirochetes. So as you can imagine, we're not going to work with any of those in this lab. Right? And they're not easy to grow and keep happy. There are others that live in the ruminant of cows that help them digest the cellulose that they eat. And then, of course, we always focus on the bad stuff, right? But microorganisms, as most of us know, right, they talk about probiotics all the time nowadays, right? That actually can be quite good. And this tribe in South America has the most diverse microorganisms associated with their skin and their intestines. And they actually found a microorganism associated with them that prevents kidney stones. So it is actually healthier to have a more diverse, and that's again why they're promoting all these probiotics and stuff like that, to try to diversify the microbes in your gut. We are actually at a disadvantage in the developing world because we limit our exposure to microorganisms. Yeah, that helps us not get the bad ones, but we're also not getting exposed to the good ones. So as I said, Staphylococci and Staphylococcus Epidermis, as you can imagine, gets its name that it's commonly found on our skin. And it forms clusters and even can form biofilms and matrices where they really stick to each other. So they can cause some pretty serious problems when they get where they're not supposed to. Some examples of streptococci, as I said, Streptococcus pneumoniae causes strep throat, right? As well as some strains of it can produce toxins that cause things like scarlet fever and toxic shock syndrome. Streptococcus pneumoniae, this is a really nasty one as well. Can cause really serious form of pneumonia, hence its name. But it can also, when it gets in other places, cause pretty serious infections. Vibrio cholera, as I said, is the one that most people are familiar with, that can cause pretty serious infection. And this other Vibrios is one that we worry about here in Louisiana because it can contaminate oysters and cause infection. And this infection was actually from someone in the waters and had cuts, little cuts, and it got into the wounds. So today we're going to do two different staining techniques. We're going to do simple stain and negative stain. Each person is going to make their own 4 slides. They're going to simple stain 2 of them and negative stain 2 of them. Each of our set of slides is going to have a cocci and a rod for us to look at. So these stains work because bacterial cells have an overall positive or negative charge. No, they're negatively charged. They're negatively charged. And most of the dyes that we use are basic dyes. So some examples of ones are crystal violet, methylene blue, and safranin. We're going to use the first 2 today. These have a what charge? These are positively charged. So they're going to stain the cell because like opposite charges are attracted, right? So the plus and the minus attract each other. And so instead of the background being stained, as you can see in this picture, the cells themselves are being stained. When you use basic dyes which have a positive charge, they're attracted to the negative, the opposite charge of the cell. The opposite, of course, is true here in this picture, right? The background is stained. So we're using a different type of dye. We're using an acidic dye. This picture is using nigrazine, which we'll use today. Congo red, which is another dye, red dye that we're going to use today. It's also an acidic dye. And an acidic dye that we don't have but is mentioned in your homework is eosin. All of these have what charge? They have a negative charge. So the cell's negative, the dye is negative, so they actually repel each other. And that's why the background gets stained instead of the cell. So as you can see here, right, what shape is this? Rod, and it's forming a chain, so we call that Streptobacillus, right? Here are cocci forming groups of 4. These are known as Tetrads, and your Micrococcus luteus should look just like this today. It should look just like this picture. So why are cells negatively charged? Anyone know what this represents, what molecule? So you have a sugar nucleotide base, a phosphate. This is DNA, right? Or RNA. Look at all these negative charges. It's a highly negatively charged molecule. And then the membranes are made out of phospholipids. Right? So again, lots of negative charges there that attract the positive components or repel the negative components of acidic dyes. So what's the consequences of leaving a stain on the bacteria smear too long or overstaining? Well, think about this. When we simple stain with a basic dye, the dyes are going inside the cells, right? And attaching to the membrane. So it's almost like you're filling up a water balloon, right? So what happens if you leave that stain on there too long? You could pop it, right? Or you could definitely distort it, which would make it hard to see. Especially if you have like Streptococci or Staphylococci, it would be hard to see that because they kind of blur into each other. So you don't want to stain too long. The golden rule in microbiology is 1 minute. Notice the clock in the back has a second hand, right? That's the little red hand that goes around, as well as I need to move my boxes so we can see our other clock. Right? So you don't need to take out your fancy cell phones, but you can, right? But you can also just look at the clock. What if we don't leave it on long enough? What if you only do like 10 seconds because you're impatient? Yeah, you're not going to be able to see it. The whole reason why we're staining is to be able to see it. If you don't leave it on long enough to stain it enough, you're not going to see it. So the next talks about surface to volume ratio. And why is this important? Is it different for different shapes? Well, absolutely, right? A rod shape and a sphere of the same volume have a different surface area. So we did the math out for you. And a typical rod has about 26% more surface area than a sphere of the same volume. Right here is about the upper limit for a rod-shaped bacteria. They don't really get much bigger than this. And they really don't get flatter, right, where they're kind of like a disc. But what one of our cells is kind of like a disc? And it probably would make sense because it creates a lot of surface area. Red blood cells, right? They're actually biconcaved even, right? And again, that's to create a lot of surface area as it relates to its volume. So we're going to do a little activity. This is of Play-Doh turned one into a rod and one into a cocci. So it's the same volume, right? If they're the same size pieces, you can see that that rod has greater surface area than your cocci. Now you can squish your two pieces together. And you can have a little bit more fun. What other shapes can we make? What other shapes are there? Granted, we're only going to see cocci and rods in this class, right? But what's this one? Vibrio. See how talented you guys are. See if you can make the other ones. Yeah, you could make a chain. I bet you I can do it better. It's basically a long rod, right? And then we... Right, so there's your three-dimensional spirochete. And if I kind of flatten it out and make big curves... Looks like a snake. What would that be? Spiralium. And again, both Spiralium, Vibrio, the spirochetes, all have a greater surface area, right, than our poor little cocci. And that's why our cocci are really small, because as they get bigger and bigger, they don't have enough surface area to support their volume. So why is this important, though? Why is surface area compared to the amount of volume important? So you can keep playing if you want, or you can ball it up and put it back in your container. Why is that important? Why is this? What happens at the surface of the cell? What defines the surface of the cell? What layer do we call that, at the very least? You have a cell wall, but every cell has a cell membrane, right? What happens at that membrane? Stuff goes in and out. Transport, active transport, facility, all that fancy stuff, right? But if you're a single-celled organism, stuff's got to get in, food, and waste have got to get out. that all happens at the surface, right? So within the surface, there are embedded proteins that facilitate, right, the stuff going in and out. If you don't have a lot of surface, then you can't support as well your volume and that need for stuff to go in and out. And then it creates even more of a problem. Make sure you seal them up good so they don't dry out on you. If you have limited, say, doors, right, like us, we've got one. We're getting the heck out of here. Think about, again, that surface area. And that even relates to our food, right? Which one of these is going to cook faster and why? This one, you know, this hamburger, or this meatball, right? Which one are you going to have to cook the longest? That meatball, right? Because you got to wait for the heat to penetrate all the way through. Where if you spread out the hamburger meat, right, in a patty, it's going to cook much quicker. Same is true for the sausage, right? The thinner sausage is going to cook faster than this really thick guy, right? My husband boils these before he puts them on the grill, right? Do other organisms have to deal with surface to volume ratio issues? Yeah, good old trees, what do they do to increase their surface area? Leaves, the broad, flat leaves. That's why leaves look the way they do, right? And then even the needles are like little solar panels. But again, they're increased surface area. So, Cox's has got a problem, right? He's tiny, right? The rod has got him beat. But, who's going to survive in a dry environment better? The Cox's, right? Because what do you have to worry about losing in a dry environment? Water, right? And water's precious. All living organisms need it. So if you have a small surface area, you're going to lose less water, right? So a cocci is actually better adapted to a dry environment because of that smaller surface area. But if you're in a nice, moist environment, who's going to win? The rod, because he has the greater surface area, right? He's going to grow faster. He's going to be able to take in nutrients faster. And he doesn't have to worry about losing water because the environment's moist. Why doesn't a negative stain colorize? Why doesn't a negative stain colorize the cells in the smear? Right, so negative staining, we use what type of dye? An acidic dye, which has what charge? And cells are negative. So cells negative, dyes negative, they repel each other, right? The background gets stained. So eosin is an acidic dye. I think they just tell you it's red in the book, right? So I had added that to the homework. Methylene blue is a basic dye that's blue. So based on that information, if you were to stain a slide with eosin, what would get stained? The background, because of the negative charge in the negative cell. And then, if you stain it with the basic dye, what gets stained? The bacteria, the cell, right? So you would have a blue cell with a red background, right? I honestly have never tried this one. But also depends on the order, and that's some of the things that you'll think about as you do the homework for next time, because we're going to start combining stains, right? And it'll make sense on what gets colored and why we do it. Okay? So unlike in pre-K when you would mix red and blue and get what? Purple. That's not the case here. Okay. So the other difference between these two staining procedures is that when you do the basic staining procedure, right, with the simple stain, with the basic dye, what are we going to have to do first before we can stain those slides? What do we have to make first? What do they say you have to make first? A smear, right? And in order to make a smear, we're going to put bacteria on a slide. We may need to add what to that slide to be able to spread the bacteria around? Water. Okay. And then what else do we have to do to the slide? We have to heat fix it, but before we can even heat fix it, we got to let it air dry. And we're going to speed that up by putting them on our light boxes, which should be getting nice and warm now. When we heat fix, the reason why we do that is to cause them to do what? Stick to the slide. You will kill them, but so will staining, right? That's not why we heat fix them. We heat fix them to stick them to the slide. What happens to them when we heat fix them? Are they going to stay exactly how they were before we heated them up? No, they're actually going to shrink a little bit. So if you were to compare one that we simple stained where we had to heat fix to the exact same cell that we did a negative stain where we do not heat fix, the ones that we heat fix are going to be smaller because the heating will shrink them. And so this is the other important thing. Think about before non-stick frying pans, right? If you fried an egg, what did you have to stick in the pan ahead of time? Butter, some type of lubricant, right, to keep it from sticking. Cells are made out of a lot of protein. So is eggs, right? A lot of protein. Proteins become sticky when you heat them. That's why an egg will stick to that pan. Right? So when we heat these cells, they become sticky. And then also envision, right? Frying up that egg, it kind of shrinks up, doesn't it? So again, the proteins coagulate, they shrink up when you heat them. So think about frying that egg as you're frying your bacteria today. You're sticking them and you're shrinking them. Right? And it's that heat fixing step that we've got to do for simple staining with basic dyes that we don't do for negative staining with acidic dyes.